Well, good evening, everyone, and welcome to the George Washington University. My name is Dennis Narango. I'm Associate Vice President for Development at GW Medicine. It's a pleasure to have you join us at the School of Medicine and Health Sciences for our latest edition of Frontiers in Medicine. The Development Office organizes these events in collaboration with our department chairs and faculty leaders to bring you the very latest information about some of the most fascinating topics in the health arena and involving the future of medicine. The physicians and scientists here at GW work every day on some of the most complex and vexing problems that affect us, both in the laboratory and in the professional setting with patients. And they do amazing work. Tonight's topic is accelerating neuroscience to neurotherapy, and I think you'll find it quite fascinating. Now, our featured faculty tonight include some highly outstanding and accomplished individuals whom you'll meet in just a moment. They include Dr. Henry Kaminsky, who's the chair of Department of Neurology, Dr. Mohamed Kobesi, also of the department, Dr. Anthony Lamontia, the director of the GW Neuroscience Institute, and Dr. Vittorio Gallo, a member of that institute, as well as Children's National. We'll have a series of short presentations, and then there'll be an opportunity for you to ask questions. And those of you, I see many faces who've been here before, so you know the routine on the questions, and staff will be coming around to get them. It's a privilege now to introduce our moderator, Dr. Henry Kaminsky. Dr. Kaminsky is the Meta A. Newman Professor of Neurology and Chair of the Department of Neurology. He's also Professor of Pharmacology and Physiology. Dr. Kaminsky is an internationally recognized expert in the care of patients with myasthenia gravis, which is an autoimmune neuromuscular disorder that causes muscles to tire and to weaken much more easily than they should. Dr. Kaminsky is the author of more than 100 peer-reviewed articles and book chapters and serves in several national leadership roles, which I won't bother to delineate because it'll take a little too long. But this is a highly accomplished leader in the field. Before joining GW Medicine last fall, Dr. Kaminsky spent most of his career at Case Western Reserve University and also served as chief of the neurology service at the Cleveland Veterans Affairs Medical Center. Ladies and gentlemen, please welcome Dr. Kaminsky.
Thanks. So thank you very much, and welcome to Accelerating Neuroscience to Neurotherapy. I'm going to just take a few minutes to dissect that title. So neuroscience is trying to study the function of the brain and the nervous system as well. And I can't imagine anything more exciting to try to understand. It is, after all, ourselves that we are trying to understand as neuroscientists. But neurotherapy is really what touches everybody here in the room. I cannot imagine a human being who has not been affected by some neurological disorder, whether it may be as simple yet devastating as migraine headaches to something much more profound and debilitating like Alzheimer's disease or epilepsy or multiple sclerosis that you'll hear a little bit more about. And that's what we really want to hear about, new therapies. And so that moves on to the accelerating piece. And I'm not quite sure what that means. I think it is the process by which we do things. And I hope at the end of this, maybe we'll have an idea of how we can try to accelerate that. But I'd like to give you two examples, one personal one, about what acceleration is or what the speed of research is. So about 20 years ago, I started studying this disease, myasthenia gravis. It's a rare disorder. 200,000 people in the country have this. It can be devastating. People can become extremely weak, be on a ventilator. They can develop impairments of eye movement, so they can't see, can't function. So it's a highly debilitating disease. And about 10 years ago, after really becoming educated on how to do research, I developed an idea about therapy. And only now, after 10 years, we might be getting into a clinical trial to test the first agent for myasthenia gravis out of my work, and we continue to try to improve on that. But what I want to get into is that that's 20 years, and my patients are still waiting. You know, I'm still seeing these people come into our hospitals here and into the clinics who are debilitated by this disease. So I'm deeply interested in how we can all work together as a society to accelerate research in neurological diseases. And I'll give you one more example before I introduce our speakers on how difficult this task is. We hear about Alzheimer's disease. We know that it's a debilitating, awful disease that affects a large number of the world's populace. We also know that the National Institutes of Health and pharmaceutical companies are trying to find therapies. Now, whether it's from a profit motive or from a deep-seated desire to try to help people, really doesn't matter in some ways. The success rate has been poor. There have been 800 compounds that have been tested and found to be efficacious in mice, and none of those have gone to humans. So that tells us how difficult this process is. And so with that, I'd like to turn to our speakers and what they're going to be talking about. And they're going to be telling us about their work and their lives in the neurosciences and how they're moving on to neurotherapeutics. I'm going to take a brief moment to introduce each of the three speakers to try to move the program along. You really want to hear what they have to say. Dr. Anthony Lamontia is our first speaker. He's the director of the Institute for Neuroscience here at GW and was brought to GW to establish one of the premier programs here on the East Coast in studying various aspects of neuroscience. We have a close colleague, Vittorio Gallo. He sits and does his work at Children's National Medical Center, one of GW's close partners, and he is world-renowned in his studies of demyelinating diseases and the function of myelin, the coverings of the nervous system. And then finally, we have a new person that we brought to GW, Dr. Mohamed Kobiesi, who will be talking to you about epilepsy and some therapeutic methods that he's developed to really try to help the most devastated individuals with seizure disorders. So with that introduction, I'd like to have Dr. Lamontia come up and begin the session and have our other speakers sit down so they can enjoy your talk as well.
Okay. So as everyone is getting settled down, Henry has asked me before I start my scientific remarks to tell you a little bit about my own journey as a neuroscientist. And so what I will tell you is that I became a neuroscientist when neuroscience was first emerging as its own field. And I think that's important for this idea of accelerating neuroscience to neurotherapy, because the accelerant for therapies in any disease is the underlying science. So I was awarded the first PhD degree in neuroscience from Yale University. I'm a co-author of the first textbook that's truly an integrated neuroscience textbook for training medical students in the neurosciences. And in my career, I've been able to, thankfully, integrate the cutting edge of molecular and genetic technologies with questions about the nervous system, its function and its dysfunction. And that's what I want to tell you about this evening. So if we are to understand any disease and begin to develop therapies that will be incisive and provide comfort and recovery for patients, we actually have to understand the underlying cellular causes of those diseases. And if you think about it this way, for understanding cancer, an effort was begun in the 1940s and 50s to ask what went wrong. It was eventually recognized through years of cell biological and molecular and genetic analysis that cancer was a disease of many organs, but underlying the whole problem was that it was the way that cells divide, the way that they move, and the way that they die. We are on the threshold of beginning to understand, similarly, what's the foundational cellular problem in many, many diseases of the nervous system, many of which Henry enumerated for you, and some of which I will now enumerate for you. But to begin with, you need to understand the target. And just in the way that we understood finally in the 50s that for cancer, the underlying target was cells, how they divide, move, and die, we now understand that for behavioral disorders, for disorders of the nervous system where behavior no longer functions properly, the cerebral cortex is the target of pathogenesis. That's the part of the brain where things go wrong. Now, the cerebral cortex in this MRI image is all of this huge expanse of neural tissue here. There are other parts of the brain, true, but it's this part of the brain that is compromised in these disorders. So I've indicated it in yellow here, but if we just take a little look at what the cerebral cortex looks like, if you were to stain that tissue with a stain that tells you where cells are, what you see is that basically the cerebral cortex is this accumulation of all these little purple dots or cells. They're arranged in layers, and we'll have more to say about that in just a minute. And each of these cells is a single processing unit of the nervous system. And the way that they connect with one another provides a circuit to represent information and to allow us to do the things we do, whether it's my ability to talk to you and tell you this story, your ability to see me, to hear me, and hopefully to understand what it is I'm saying. So if we look at just one of these cells, one down here in the fifth or sixth layer of the cortex, what you see is here's the cell body, the purple dot. But these cells have remarkable processes. These are the surfaces onto which nerve cells make connections and receive connections. And this is an incredible tangle, you may say, but it's in this tangle that not only the appropriate functional connections are made to make a brain operate correctly, but it's in this tangle where those connections go awry and brain function diminishes. Now, I'm going to make this a lot simpler for the purpose of this evening. I'm going to divide the cortex. You can see there are these 6 layers here. The numbers are enumerated, but I'm going to divide it into 2 halves, basically. Layer 2 and 3, where there are these orange neurons that are called projection neurons, and that's because among their branches they have this long process, this axon, that's going to leave the area of the cortex where the cell's body is and go and make a connection somewhere else. Then there are these blue nerve cells that are called interneurons, and these are the neurons that say no. They only have axons, these little blue processes here, that are very local. And they make a little local network and tell the projection neurons, no, don't send information out yet, you're not ready. So this is the neuron that says yes, the orange neuron, and sends that information somewhere else. The blue neuron is the neuron that says no. And you'll see that these 2 nerve cells come from very different progenitors during development, and that's going to be important. And it's going to be particularly important to discuss these nerve cells that are in layers 2 and 3 of the cortex, because it turns out for the diseases that I'm going to tell you about, it's these connections made by these neurons that are thought to be the targets for pathogenesis. Now, we've recognized over the last decade that indeed faulty circuits, and this is a Scientific American article by Tom Insel, a very good friend of mine who is the director of the National Institute for Mental Health, that basically the cerebral cortex can be reduced to this circuitry, and that faults in the circuitry are actually the causes of psychiatric and other behavioral disorders. Now, what are those disorders and what causes them? The other thing that we've recognized, because we were wise enough to make the investment in the human genome and have the capacity to do human genetics of the most complex diseases, that there are distinct genomic causes, and your genome is, of course, the complement of DNA that you have in each of your cells, that cause genetic diseases. You can either have mutations of a single gene. They can be dominant or recessive, so either you have too much function of the gene or the gene doesn't work, and it can cause either a loss or gain of gene function. Now, there's also other ways to break the genome. You can duplicate genes. You can duplicate all or part of a chromosome, And this increases the amount of the gene products, the proteins that are made that are encoded by these genes. But the genes are normal in their function. You just have too much. The other way you can break a genome is you can delete genes. So basically, you can lose part or all of a chromosome. This causes a decrease in the gene product. So you haven't changed the function of the gene. You just have half as much of it. And if we look at behavioral diseases, an example of a mutation-caused disease is Fragile X syndrome, which causes mental retardation and other things, or intellectual disability. A duplication is Down syndrome. And a deletion is this syndrome called DeGeorge syndrome. Now, you may not have heard of DeGeorge syndrome, but you're going to hear a lot about it tonight because it's what my laboratory studies. And this occurs in 1 in 10,000 children, 1 in 1,000 children, 1 in 2,500 children. So it's the most common deletion disorder known, and behind Down syndrome, it's the most common example of what are called copy number variant diseases. And these CNVs, it turns out, are associated with a whole range of disorders that I'm now going to tell you about. So we're going to use DeGeorge syndrome as our model for understanding why it is that half is really not enough in this case and in the case of building neural circuits. Now, what I want to argue is that there's a whole set of diseases that you may not have thought of as related, but they can all be grouped into this umbrella of diseases of cortical connectivity, just in the way that we can group cancer as diseases that cause changes in the ways that cells divide, move, and die. Now, diseases of cortical connectivity include autism, schizophrenia, mood disorders, including bipolar disorder, depression, language disorder, social cognitive deficits, intellectual disability, attention deficit, hyperactivity disorder, and schizophrenia. So this is a broad range of disorders, but when we look at them, what we recognize is that they all share a common thread, and that is that they're disorders of the way that cells in the cerebral cortex make connections with one another. The reason for these disorders is that in each case, there are genetic lesions. There is something that goes wrong in the genome, either a mutation, a duplication, or a deletion, or a combination of all, that actually lead to disruptions of development. So the hypothesis that has gained favor, and that we're going to test in a very specific example, is that all of these diseases are caused not by the brain being built correctly and then running down, but by the brain never being built right to begin with and operating incorrectly throughout the lifespan. That leads to aberrant circuits that process information in a disrupted fashion. And so therefore, I'm going to argue that these diseases of cortical connectivity, all of these disorders, are most likely genetic diseases that disrupt the development of that cerebral cortex that I showed you. Now, you need a genetic lesion to start to study this. And DeGeorge syndrome is a specific genetic lesion. And what it is a deletion of a small number of genes on human chromosome 22. So this makes it a model of copy number variant disorders. Now, what you see here is a chromosome smear. The 2 dots here are 2 copies of a gene on chromosome 22 that's outside of the deleted region. You see only 1 dot here, and that's a marker for a gene that's in the deleted region, and there's only 1 copy. Now, what happens in this disorder is very simple. On the chromosome 22, from either the mother or the father, one of the 2 copies has lost a small piece of DNA. And this piece of DNA is relatively small. It's about 1.5 megabases, which is just the way we measure DNA, but there are about 30 genes here. So not many genes when you consider that there are 25,000 genes in the genome. So just having 1 copy rather than 2 of 30 genes can cause a lot of trouble. Now, the typical deletions are a little bit larger, but this deletion gives you all of the phenotypes, the disease phenotypes. The correlation between the genotype and the phenotype is variable, which means that it's unlikely that only one gene in this region causes the disorder. And this is the most common deletion disorder known, 1 in 3,000 live births. It's now been revised down to 1 in 2,500, which is the number I gave you before. These kids have a distinctive appearance. Their foreheads are high, their eyes are wide set, their brows are flattened, their nose is bulbous and turned up at the tip. They have a long philtrum, they have a diminished upper lip, a protrusive lower lip, their chin is diminished, and their ears are low set. They have craniofacial disorders. But beyond that, they also have heart malformations, which used to be fatal until corrective surgery for infants came along. They have limb and digit malformations, but most importantly, there's an increased incidence, almost to 100%, of major behavioral disorders, including schizophrenia, autism, ADHD, and bipolar disorder. In other words, those disorders of cortical connectivity. So something about this loss of one of two copies of these genes causes a change that makes the brain susceptible to this range of disorders. And what we're going to try to do is we're going to try to figure out why that is and if that reflects altered development of the brain. What we know is when we look at the mature brain, the actual conformation of gray matter and white matter in patients with 22q11 disorder changes. So the cerebral cortex is different. But what we have to understand is whether or not that difference arises during development or whether it arises as a degenerative process. So we can actually not study development in humans because by the time we know the disease has happened, the child has already been born and most of brain development is concluded. So we need to study it in an animal model, and we can do it in the mouse because the mouse has roughly the same genes all in a row. And what we can do is we can make genetic models by actually engineering the mouse genome so that the mouse has a deletion that's exactly parallel to the deletion that we see in humans. So it has half the number of copies of these genes, just like its human counterpart. And we can study development from the beginning of making the brain to the very end and actually ask whether or not having half of the dosage of these genes has consequence for how you put together those circuits in the cortex that process information. So what you need to know to follow the rest of this story is that when you build a cerebral cortex, when you make these cells, they come from a distinct kind of cell that can divide, because nerve cells themselves, once they're made, they never divide. So you need a stem cell or a precursor, and that precursor is called an apical precursor or a radioglial cell. And in order to make the cells, particularly these projection neurons, it makes an intermediate dividing cell that's called a basal precursor. And this cell divides a lot, and it divides rapidly, and it makes enough of these cells so that you have the right numbers in the cerebral cortex. And now what I'm going to tell you is that these basal progenitors, but not these apical progenitors, these basal progenitors don't divide right. They don't make enough projection neurons during development when there's only 1/2 of the copies of the 22q11 genes. And what we see is that, yes, indeed, in the cerebral cortex, the number of cells in layer 2 and 3 is diminished by about 20%. These are those orange-coded projection neurons. So in layer 2 and 3, you don't have the right number of the cells that are taking in information and sending a signal to the next place for further processing. So that means that loss of one of the two copies of the 22q11 genes causes this cell during early development not to know how to divide properly. That disrupted ability to proliferate, the stem cell is broken, its progeny don't show up in the right numbers, you lose some. And so part of this important circuit for processing information is broken by virtue of this genetic lesion. So it disrupts the capacity of the cortex to put together circuits that process information. Now, what about the other cellular element of this circuit, the GABAergic interneuron? Well, what you need to know about these cells is something really surprising. Even though these cells live in the cerebral cortex, they're not made in the cerebral cortex during development. They actually have to migrate in from another place. So they are actually made elsewhere in the brain, and they have a long, strange journey where they actually crawl through brain tissue and end up in either layer 2 and 3 or layer 5 and 6. So these cells not only have to be made, they have to know where they're going. And that gives us another place where you can break the system. Now, what happens in the 22q11 deletion is I'm going to show you here. We can actually watch these cells begin and make their journey. They start down here in the bottom of the brain. We've labeled them genetically, and we're now going to show a movie. And this movie is going to flip because of oddity and projection, but you'll see the point. So if you'll start the movie, please. There it goes, it flips. But you see these cells are moving, and they're moving into the cerebral cortex here. This is the normal. You see they've moved all this distance. Now play this movie, please. This is the large deletion. This is the DeGeorge mouse. You see they're moving slower, and they don't move as far in the same amount of time. And if I was to show you the difference graphically, this is the difference. But we can actually measure the velocity of individual neurons. And what we can show is they move 25% slower. Now, remarkably, you make the same numbers of these cells, but when they have to move from where they've made to the cerebral cortex, where they're going to take up residence and do the job of processing information, they don't move there as fast, and they end up in the wrong places. Now, we can show that they end up in the wrong places because we can actually count the numbers of progenies. And what happens is that in layer 2 and 3, you normally have fewer of this particular class of cells, but in the large deletion, you double the number of cells in layer 2 and 3 of GABAergic interneurons. So now we have a real picture emerging, because in layer 2 and 3 of the cortex, when you don't have the right number of 22q11 genes, you only have one copy rather than two, you diminish the number of projection neurons, and you increase the number of GABAergic interneurons. So you have too many cells saying no, and not enough cells saying yes and projecting out to provide information. It turns out that the reason that that happens is these cells are molecularly distinct. We can sort them out. We can interrogate all of the genes they express. And we found one gene in particular that was really interesting. This is a receptor for a cytokine. These are molecules that are made by the immune system. And this receptor is actually not present. It's about 50% diminished because the 22q11 genes are missing. And what that leads to is it actually means that when the cells migrate, they don't migrate in the right spots. And it's this migration change, and you can see this here, and we've actually tested this genetically by taking one more copy of that receptor away. And you can see how the cells progressively get more and more lost the more you change the system genetically. So what that tells us is that by missing one copy of the 22q11 genes, you change the cellular elements that make the circuits of this layer 2 or 3 of the cortex, where important information processing happens. There are fewer of these cells, these projection neurons, that say yes, and there are more, a change in migration that causes a switch in the frequency of the cells that say no, so that there are more up here and fewer down here. So this actually tells us that, yes, indeed, it's likely that when you start with a genetic lesion, that you actually change the cellular construction of the cerebral cortex, and then that can be the underlying reason for these grave disorders that start early in life and compromise an individual's ability to lead a life and compromise their family's ability to help them launch on the best life possible. Now, the last thing I want to tell you very briefly is that this has, in these mice, as in humans, consequence for behavior. We can actually train mice to make very specific cognitive decisions on which visual stimulus is going to give them a reward. Then they have to change the rule. So these mice have to know, this is what I need to do to get my food reward. And then when the rule changes, they have to be flexible enough to change the rule. And we're not going to look at the mouse doing this, because that'll take too much time, but they do this. And what we can show is that The large deletion mice, the 22q11, the DeGeorge mice, have really, really altered cognitive flexibility. They make far more errors in learning this task, and it takes them a lot longer to learn the task. So this tells us that their behavior is different, and we know from other work that the circuitry that's really critical for performing this task is focused in layers 2 and 3 of the cerebral cortex, particularly in anterior or frontal regions. So, to wind up, what I think this tells us is that I've shown you that 22q11 deletion syndrome, or DeGeorge syndrome, as it was first called, is a copy number variant-related disease of cortical connectivity that arises during development. The brain is broken when you put it together. It never functions properly because the cellular elements are not arranged in the right pattern, making the right connections. This is caused because of a minimal deletion of 1.5 megabases, 30 genes, on one chromosome, leaving only one copy of those genes. It disrupts development. It leads particularly to autism, attention deficit hyperactivity disorder, and schizophrenia in these patients. There are other genes that modify, and also environmental factors that can modify how significantly development is disrupted. We've shown this in my laboratory, but I'm not going to tell you about that work. And the result is aberrant cortical circuits. So we know a little bit more. We know that this genetic lesion actually causes a cellular change. And in a sense, this is equivalent to the sort of information that we needed to begin to understand cancer pathogenesis. And colleagues in other laboratories across the country working on other genetic disorders are doing similar studies. And it's all emerging that how you put together the cerebral cortex, how you make the cells, and how they move around is where the system goes awry. And now we have to understand better what that results in terms of behavioral pathology, and where we can target the process, either during development, which is really a novel sort of therapy, or once development is completed, so that we can ameliorate the developmental disruption to restore adult function. So with that, I'll thank you for your attention.
Is the microphone on? Okay. Well, thank you very much, Harry, for the introduction, and thanks very much for inviting me to this very timely and exciting symposium, and thank you all for coming here tonight. As Anthony mentioned, for his own career, for his interest in neuroscience, also my interest in neuroscience started when I was a graduate student. I was at the University of Rome, and I was doing my thesis work at the National Research Council. And I was very interested, starting early on in my career, I was very interested in brain growth, in brain development, and the possibility of regrowing nerve cells or stimulating regrowth of nerve cells under, in different neurological diseases. And I was lucky enough to work with a mentor, who, Rita Levi-Montalcini, who was the first person who discovered one of the growth factors that stimulate brain growth, growth of cells in the nervous system. And Rita won the Nobel Prize in 1986 for her work and continued to be a very important mentor in my career. So what I'm going to do today, I'm going to talk about a different part of the brain than what Anthony has talked about. And in particular, I'm going to talk about the white matter. This region of the brain is shown here with a 3D reconstruction of a diffusion tensor imaging, diffusion tensor image. And the areas that are shown here in colors are all areas of white matter tracks. So the white matter, the brain white matter, is rich in connectivity, and these connectivities make connections between the white matter and different regions of the cerebral cortex, both the anterior cerebral cortex as well as the posterior cerebral cortex. But very importantly, there are also very important connections and connectivity between different brain hemispheres, and these reside in the white matter. So the white matter tracts are, the majority of white matter tracts are myelinated. They are wrapped by this electrical insulator, myelin, which plays a fundamental role in brain function. So myelin allows this wrap that is shown here in by electron microscopy. Myelin allows propagation of action potentials, propagation of nerve impulses in a very efficient way throughout these nerve fibers. And the velocity of propagation of these impulses is much rapid in fibers that are wrapped with this electrical insulator. But is myelin important only for propagation of nerve impulses? Or what is the fundamental function of myelin during development and for brain function? So if we look at the neurodevelopmental timeline of the human brain, what we know is that myelin is a relatively late event. It's a landmark in brain development, but it's a relatively late event. Myelination occurs in the brain, begins in the brain during the third trimester of pregnancy, and continues in the young adult, and it continues for many, many years after birth. So myelination and developmental myelination is a largely postnatal process that spans many, many years, beginning at birth into young adulthood. Now, why is myelin important? Why is developmental myelination important? There is a vast literature on the importance of myelination in cognitive development, in executive functions, and I just wanted to give you some glimpses from this literature and some examples. So, for example, we know that there is a correlation between developmental myelination and cognitive development. This has been widely demonstrated in executive function analysis and cognitive development in children and in young adults. There is also a correlation between myelination and language developments in infants and in toddlers. And I'm sure that many of you have experience with teenagers. Anybody who has interacted with teenagers in this room knows that there is a very important correlation between myelination and functional organization of the frontal lobe. Finally, there is, we know that this myelination is disrupted in several neurological disorders, and it's really a key neurobiological component underlying both neurodevelopmental and behavioral disorders. And I want to make a point that today I'm going to talk about the effects of hypomyelination or low levels of myelination during early development, but as many of you are aware of, there are many demyelinating disorders, diseases in the adults, such as multiple sclerosis or other demyelinating diseases that occur later on in life. But I just wanted to give you an example that is sort of relevant to what I'm going to talk about later on in this presentation. And this is a recent study done in premature infants in which white matter function and structure and function has been analyzed by diffusion tensor imaging by looking at a parameter called fractional anisotropy. Fractional anisotropy basically gives us an idea of the functional organization, the structural organization of the white matter. And what this study concluded, very interestingly, is that there is, in these premature infants, there is a correlation between the extent of disruption of abnormality in the white matter as measured by fractional anisotropy, and shown here with these red tracks. There is a correlation between this disruption and total IQ in these children that are followed later on in the studies. So this is just an example of how in this very vulnerable population of infants, premature infants, there is a correlation between abnormalities in their white matter structure and function and total IQ. So what are the data about prematurity in the United States? What is the extent of this population? How large is this population of premature infants who are at risk for white matter diseases and later on cognitive dysfunction. So what we know is that about 13%, these are data from 2007, but they're very similar and they still hold pretty steady in more recent years. About 13% of the total populations of newborn in the United States, that's about 500,000 babies every year, are born premature. And 2% of these are very preterm. They are less than 32 weeks. And their size is extremely small. They can fit in one of your hands, and they're extremely small. And 1.5% are less than 1.5 kilos. So this has a very strong impact, as I will articulate later on in my talk, on families and society, and the cost of maintaining these infants alive, and later on for different types of interventions in between $46,000 and $230,000 a year. Now, what are the consequences of prematurity in the white matter? So we've become very good at maintaining these babies alive. So the improved neonatal care promotes about results in about 85–90% survival of this population. However, later we are not very good at preventing the neurological sequelae, the neurological dysfunctions associated with prematurity, and particularly with white matter injury. So prematurity is associated with both cognitive and neurodevelopmental delays, and in fact, in many longitudinal studies, it has been shown that about 25 to 50% of these children or later on adolescents require special education services. 50% have cognitive deficit on IQ testing and deficits in executive functions, and more than 30% display ADD or ADHD. When followed up to the 4 years of age, about 50% of them exhibit very serious behavioral and adjustment problems compared to full-term control peers. And there are gender differences. The males experience more delays than females. So what are the consequences of prematurity at the white matter level? In general, if we look at a neonatal MRI, what we find that there is this, the most common white matter consequence of prematurity is this so-called diffuse white matter injury. This is a new term, a relatively new term, and this is a 2-year-old boy that was born at 26 weeks of gestation. This is from Children's National Medical Center, and this boy was taken care of by a child neurologist in NICU who is part of my research team, Dr. Joy Scafidi. And Joy got this MRI picture from this boy, showing this boy displayed spastic diplegia, language delays later on, sensory motor difficulties, a variety of neurological impairments. But what is very clear in this MRI image is that we found that in this boy there was decreased white matter volume, a very strong reduction in the total volume of that part of the brain that I showed you earlier on. And also there was an increased signal around the ventricles. These are the brain ventricles, and you can appreciate the signal, the increase in the signal around the ventricles. And the general signal in the white matter was diffusely abnormal. So these are the main characteristics. This is one of the main injuries that we find almost consistently in all these premature infants. And what happens later on in development? This is a study done in young adults, in adolescents, and in young adults that were born prematurely. And in these studies, the investigators of this, that published this article detected perceptual, cognitive, and motor impairments that are all associated with white matter injury. And we're actually able to associate different types of impairment, like fine motor problems or visual perception problems, with specific white matter tracts that were detected as being abnormal by diffusion tensor imaging. So we are now able to associate different behavioral and cognitive impairments with specific white matter abnormalities. What is the cause of these white matter abnormalities? The main cause is low levels of oxygen in these premature babies who have underdeveloped lungs and have oxygenation failure and white matter changes that are detected in all these preterm infants, of course, with a broad range of severity. These regions that are circled here in red represent, again, region of abnormalities in the white matter as detected by fractional anisotropy and caused by different levels of hypoxic states. So what does hypoxia do to the white matter? What is the main target of this hypoxic state in the white matter? The main target is this cell, the oligodendrocyte. This cell is shown here from one of our mice in which we expressed specifically a green fluorescent protein in these cells. And so we can map these cells directly in the white matter. We can look directly under the microscope and do 3D reconstructions of these cells in the white matter. As you can see, this cell, which is the cell that makes and synthesizes and makes myelin and wraps myelin around the axons, this cell is the main target of perinatal brain injury, of white matter injury in these premature infants. And very interestingly, very important, this cell is also one of the main target in a number of demyelinating diseases in the adult brain, for example, multiple sclerosis. And this is also the main target of one of the main cellular targets that die in the adult brain during stroke or ischemia. So we're particularly interested in understanding what are the cellular and molecular mechanisms that cause death of these cells in these premature infants, and in order to prevent death and degeneration, death of these cells and degeneration of myelin. So as Anthony talked about a mouse model, an animal model, in collaboration with a group at the Child Study Center at Yale, we created a mouse model of neonatal brain injury or prematurity. In particular, a mouse model of hypoxic-induced degeneration of white matter in the neonatal brain. What I told you so far is that in premature infants with very low birth weight, we find decrease in brain size, we find an increase in the ventricle size, ventriculomegaly, and also we find a decrease in white matter and gray matter volume. And this is shown here comparing the term with the preterms. So in the animal model of hypoxia, we reproduced all the features of white matter and brain injury found in humans. We see lower brain weight, decrease in brain weight, ventricular enlargement, as shown here in this larger size of ventricles than in controls, and also a decrease both in white matter volume as well as in gray matter volume, in cortical volume, which is the region of the brain that Anthony talked about earlier on. But we can also reproduce different phases that occur in the premature brain after premature birth. So we reproduce an early injury phase, which is 1 to 3 weeks after hypoxic injury in these neonatal mice, in which we reproduce oligodendrocyte death, a decrease in myelin protein expression and delayed myelination, and in which the mice display also abnormal behavioral tests associated with white matter function. But when we look at later on after injury, we also see a phase of apparent recovery, 3 to 7 weeks. And very interestingly, this phase of apparent recovery is also displayed by these premature infants. These premature infants, depending on environmental stimuli, depending on the extent of the injury, they do improve, then they do get better. However, as compared to the full-term babies, they never perform to the same extent as the full-term babies. So also in the mouse model, we see this phase of apparent recovery. So but how do we move now from understanding these two phases and from understanding the impact at the cellular level, at the structural level of the white matter of prematurity and early hypoxic injury, how do we move from this to identifying therapeutic targets that could be potentially utilized to promote nerve growth and regeneration of the oligodendrocytes after hypoxic injury? we have been interested for a long time in understanding cell cycle regulation. We have been interested in understanding how cells, early progenitor cells that generate oligodendrocytes, mature oligodendrocytes, are regulated intracellular at the molecular level in dividing and eventually differentiating and generating oligodendrocytes. And particularly, our previous work identified this protein called P27 and one of its targets, CDK2, as an essential regulator that promotes oligodendrocytes differentiation and oligodendrocyte growth. So we wondered whether by altering, whether the levels of P27 were altered in these brains of these mice that were exposed early to hypoxia. And what we found, very interestingly, was that P27 levels were significantly decreased after hypoxic injury. And particularly, this was due to a decrease in other proteins that regulate P27 stability, and SCIP2, a protein that regulates P27 degradation. But the important message to take home here is that P27 levels were significantly reduced after hypoxia. So a group of people in my team wondered whether by altering expression of P27, by forcing re-expression, increasing the level of P27 during the hypoxic phase, during the injury phase, would promote regrowth of oligodendrocytes. Can we make new oligodendrocytes by specifically targeting P27? And we did that by injecting a virus that carries the P27 gene and injecting this virus in the white matter. And after we injected P27 into the white matter and we compared the virus that did not have any new genes inserted in it and a virus that overexpressed P27, we found, very interestingly, to great surprise, that we could make new oligodendrocytes that we identified based on a number of molecular markers. We could make new oligodendrocytes in these brains after hypoxic injury. So P27, it's a very interesting and new putative target, and we're very actively working on this that would promote oligodendrocyte regeneration after neonatal hypoxia. But what are the target cells? What kind of cells do we need to target within the brain in order to promote oligodendrocyte regeneration and eventually functional recovery? How do we identify these target cells? And most importantly, are these cells present in the human brain of these babies, and are they present also in the mice after hypoxic injury? Well, we found a very interesting cellular target when Joy Scafidi, the child neurologist I mentioned earlier on, analyzed the brain of one of these poor babies who had died because of hypoxic injury, ischemic hypoxic injury, right after birth. And what he found was that there was a particular cell type that expressed a receptor for the so-called epidermal growth factor, one of these factors that promote neural cell growth and regeneration. And he identified this cell. This is in the white matter of this the brain of this baby that died of hypoxic ischemic injury. These cells, the EGF receptor positive cells, were cells that we identified as being part of the oligodendrocyte type, immature oligodendrocyte type, because expressed a marker called OLIG2, which is only expressed in oligodendrocyte lineage cells. So Joy wondered whether by acting specifically on the growth of these cells, by targeting these cells that express the EGF receptor, we would be able to use this as another therapeutic target to promote regeneration of these cells. And he turned on to the mouse model, and he used a molecular intervention in which he intranasally injected epidermal growth factors during early phases of injury to determine whether injection of epidermal growth factor would promote regeneration of oligodendrocytes. So I'm sure that all of you are wondering, how is it possible that by injecting this intranasally in the mouse, this would actually promote oligodendrocyte regeneration? Well, this actually happens, and has been shown for different growth factors that this is a good way to administer this. It's a clinically feasible and overcomes the difficulty of penetrating the blood-brain barrier in the brain for drug delivery. So we can inject epidermal growth factor directly intranasally, and with different types of retrograde mechanisms, this epidermal growth factor will reach the brain. I'm not going to get into the details of what these mechanisms are, but this is an accessible way to treat these pups, these mouse pups, right after brain injury. So first of all, I want to show you evidence that this actually works. So when we inject this epidermal growth factor, the HBEGF, this stands for heparin-binding epidermal growth factor, and we did 7 injections within 3 days in the early phases of injury. Injections of the epidermal growth factor this factor reaches the white matter. As shown here, minutes after injection, you can see 5 minutes, 15 minutes, 30 minutes. This is a molecular detection system that allows us to selectively detect in the tissue epidermal growth factor after injection. Not only there is an increase in the levels after injection of the epidermal growth factor, but this epidermal growth factor also activates the receptor, so it's functionally present in the white matter. So what are the consequences of administering epidermal growth factor after injury? So first of all, what Joy found is that if we compare the hypoxic brains with the brains treated with epidermal growth factor, there was a complete recovery in the number of oligodendrocytes. So oligodendrocytes don't die anymore as they did after hypoxic injury, but after epidermal growth factor, there's complete protection and the number of oligodendrocytes is normal. Not only this, but when we do diffusion tensor imaging analysis in the brains of these mice, this is the hypoxic saline. As you can see, the signal in the white matter, this is in corpus callosum, is significantly decreased after hypoxia. But in the mice that were treated with epidermal growth factor, there is a 90% recovery when we looked at different brains in the signal in the white matter, indicating that injection of the epidermal growth factor completely restores structural integrity in the white matter. But does this restore also functional integrity in the white matter? And we did some behavioral tests associated with white matter function. This is a behavioral test called the complex wheel. So what we do, we take the mice and we put them on a wheel that has 36 rungs, and they train on this wheel. And this is really associated with locomotor activity and coordination of locomotor activity in these mice. So it depends on white matter function. And as you can see, they all train equally. The hypoxic saline and the hypoxic treated with EGF compared to the normoxic. But then when we shift these mice from a wheel that has 36 rungs to a wheel that has 18 rungs, the mice have to retrain. They have to get used to the new wheel. And as you can see, the normoxic saline and the hypoxic EGF, they normally, injected with EGF, they normally retrain and reach the same velocity that they did with the normal wheel. However, the hypoxic saline, they never reach the same velocity as the normoxic or the hypoxic treated with EGF. So EGF promotes functional recovery also in this behavioral test. And in this other behavioral test, which is called the inclined beam walk, we look at foot slips. We look at how many foot slips on a 2 centimeter or 1 centimeter wide beam these mice can make during the test. And as you can see, the hypoxic mice make a lot more mistakes, a lot more foot slips. They don't walk as well as the other mice. But when we inject these mice with the HBEGF, they're again normal, they're similar to their controls, or there is a significant decrease even with a more challenging 1 centimeter width of the walking beam as compared to their controls. Now, very importantly, and this is the last point that I want to make, this treatment was performed during the age of P11 and P14. And this is during a specific developmental time window that corresponds to the early phases of the injury. So we wondered whether if we treated these mice later on after this critical developmental time window, what happened. And we found that if we treated the mice between postnatal day 18 and postnatal day 21, the treatment 7 days after injury did not result in improved performance. So there was a critical developmental time window for intervention with EGF in order to restore function of the white matter. So what I told you today is that normal developmental myelination is crucial for mature brain function and for cognitive development. There is a critical developmental time window for oligodendrocyte and white matter vulnerability in premature brain injury. The animal models also allow us to study cellular and molecular mechanisms of injury and to identify targets of therapeutic interventions, for example, cellular targets that are also present in the human brain. And I showed you two examples of two therapeutic putative targets that we're intensely working on, P27 and EGF receptor expressing cells that promote timely oligodendrocyte regeneration and myelination after perinatal hypoxia. So I think that our animal and human studies point to a critical developmental time window for therapeutic intervention after injury. And this can be really important in terms of promoting plasticity in the brain of these premature infants after they're born prematurely and they're exposed to early hypoxic injury. So I just wanted to mention the name of the people we've been working with, Flora Vaccarino and Laura Ment at the Child Study Center at Yale University. This is my team, and I also want to acknowledge the support of NINDS and the National Institute of Child Health and Human Development for the work that I presented. Thank you very much.
My talk will be divided almost equally between some generalities of epilepsy, the incidence, the clinical manifestations, and its burden on the society, and then about the new research that we're bringing to GW, we're doing at GW. Okay, so we start with some definitions. Epilepsy is defined as recurrent unprovoked seizures. So a single seizure that is provoked by certain medications or alcohol withdrawal or head trauma may not constitute the diagnosis of epilepsy. But when seizures start to recur without any known provoking factor, then this constitutes the diagnosis of epilepsy. A seizure refers very generally to an abnormal electrical activity in the brain, and the clinical manifestations of seizure depend on what brain region the seizures are starting from, and they can be anything. So despite the hundreds of years of knowledge of seizure manifestations, we still see new publications about some novel seizure semiology or clinical manifestation of seizures that have not been described before. These range from just brief muscle contractions, which may affect a finger or an arm or a foot, but they can also consist of staring spells and some unusual behaviors such as bicycling or fear with panic, or they may even consist of laughing. We call these gelastic seizures, seizures that are marked by an early laughter. So the clinical manifestations can be anything. Epilepsy, as a disease, affects 1% of the population, but between 5 and 10% of people have had a seizure or will have a seizure during their lifetime. So a single seizure is experienced by maybe 1 of 10 people in some series. But epilepsy, as defined as recurrent unprovoked seizures, affects 1 of every 100 people. And if you look at all patients who are presenting with new onset seizures and you administer an anti-epileptic medication, medications alone are effective in 60 to 70% of these individuals. So the remaining 30 to 40% will need, various combinations of medications and novel therapeutic interventions, including surgical evaluation, that aims specifically at finding which region of the brain is producing the seizures and whether it's safe to remove it surgically. Intractability is defined as the failure of 2 or more anti-epileptic medications to control seizures. So despite the plethora of new generation anti-epileptic drugs in the past 25 years, we know now if somebody fails to respond optimally to 2 medications at good doses, that means that the chance of a 3rd or a 4th or a 10th medication to achieve seizure freedom will not exceed 3 or 4%. So the initial response to anti-epileptic medications in those who are newly diagnosed with epilepsy determines the prognosis to a large extent. And what do we do for patients who are intractable? If somebody is intractable, meaning they have epilepsy that does not respond to anti-epileptic drugs, we have to evaluate them for epilepsy surgery. Let's look, before we talk about epilepsy surgery, we'll talk about the causes of epilepsy. So causes of epilepsy are different in children than in adults. They are different in terms of the relative contribution of each cause. So for example, we know in children trauma accounts for 5% of all epilepsy. Vascular meaning all kinds of vascular accidents that affect the brain, including stroke or bleeds or other malformations. It accounts for 5% of all childhood epilepsies. But look here, the vast majority, 67% of all childhood epilepsies, is unknown. We call it idiopathic or unknown, and we believe it's genetic. And the genetic basis of many of these epilepsies is yet to be disclosed. In the elderly, adults 65 years of age or older, the relative contribution of each one of these factors is different. Tumors now account for 2.7%. The vascular is 1/3, because stroke becomes very common, in individuals 65 years of age or older. Degenerative brain disease, including some of the conditions associated with dementia, also account for a nice slice, and still almost half is unknown. So regardless whether we know the etiology or the cause of epilepsy or not, which in the majority of patients we don't, the treatment will not significantly differ. Here's the incidence of epilepsy. Incidence refers to how many new cases of epilepsy are diagnosed per year in the United States. You can see that the incidence is very high during childhood. This is age 0, this is age 20, is very high. And then in early adulthood, it declines, and it stays low until after age 60, when it escalates again. We call this a bimodal distribution. So seizures are most likely to occur. They can occur at any age, of course, but they are most likely to occur in childhood and in the elderly. So how do we diagnose epilepsy? The clinical history is very, very important. One of the main questions we ask patients who are presenting with possible seizures to the clinic is, what is the earliest symptom? The earliest symptom, or the aura, or the warning sign, tells us a lot about where seizures are starting from in the brain. For example, if the earliest symptom is a twitch in the hand, we know that, let's say twitch in the left hand, we know that the area of seizure onset is very likely to be the movement region of the right hemisphere, okay? Because each side of the body is controlled by the opposite side of the brain. If it's, let's say, at an abnormal taste, we know that the seizure onset zone can be, for example, in a region deep in the brain that we call the insula. If the aura is, on the other hand, some visual like flashes of light, we know that it's in the back of the brain, in the occipital cortex, which is responsible for processing vision and so forth. So the auras, description of the auras is crucial to the epileptologist to figure out which part of the brain the seizures are starting from. And then sometimes, because seizures result in alteration of awareness, and the patient herself or himself cannot tell about their seizure manifestations, we ask witnesses. Also, the post-ictal symptoms, meaning the symptoms that follow the epileptic seizure, can be of localizing value. For example, if somebody has a weakness on the left side of the body after a seizure, we know that the seizure probably originated from the right brain, and so forth. So the history is crucial. And then we do the electroencephalogram, which is the EEG, which measures the brainwave studies, and there are certain brainwaves that are pathognomonic or specific for epilepsy, and we do, of course, imaging, MRI, in order to rule out a specific lesion that may be causing the epilepsy. And then if we find a lesion, or if we figure out which part of the brain is producing the seizures in somebody who's intractable, then we recommend epilepsy surgery or resective surgery. This MRI shows, for example, that the left temporal lobe here has been resected and it's replaced by this black, which is fluid. In somebody who had seizures for 25 years on a daily basis. And then after the surgery, he's been seizure-free for 7 years now. And we did not recommend discontinuation of medications after the resective surgery, fearing that seizures may come back, but he did it on his own, without our advice. And he stopped his medication, and he came back a year later saying, I'm still seizure-free off medications. So that confirms that the seizure focus, which was producing the seizures, was totally included in the resected brain tissue. So if we know that there is a specific lesion and we're aware that this lesion is producing the epilepsy, and we can confirm that by video EEG monitoring. So we admit the patients to the video EEG monitoring, and we monitor them 24/7 by video and EEG. The video captures the seizures and tells us how the clinical manifestations are. The EEG localizes it to which part of the brain. We put these data together with the MRI, and if they are concordant, like the lesion on the MRI is concordant with the EEG findings of where the seizures are coming from, then we recommend resection. But sometimes the MRI is negative. The MRI is totally normal, means the brain anatomically is intact, but there is intractable epilepsy. And then we have to do further studies in order to really identify the seizure focus. One of the studies is we take the patient down to the operating room, as you can see here, and we put a grid on the surface of the brain, a grid of electrodes. So instead of putting the electrodes on the scalp to record the brain activity through the skull and the scalp, we put a grid of electrodes on the surface of the brain. This grid, for example, is more than 64 contacts, and then we close the wound and send the patient to the epilepsy unit, and we record epileptic seizures in the epilepsy monitoring unit with the intracranial electrodes, with the invasive electrodes, which gives us a much better idea about which part of the brain the seizures are starting from. But not only that, these electrodes can also be used to map the brain function. That means not only do they record the brain activity, but we can use them to deliver a tiny electric current which will disrupt the brain function and tells us about what this particular brain region does. So each one of these dozens of electrodes is mapped electrically, and we can tell that if upon electrical stimulation of one contact there is a contralateral movement, we know that this is the motor area of the brain. If the patient is unable to continue reading or count or understand certain questions, we know that it has to do with language comprehension or language expression. So we know it has to do with speech, and these are regions that we certainly have to avoid, when we plan the surgery, and so forth. So this was actually one of the reasons why I went to epilepsy as a specialty, to have the opportunity to study the brain, functional specialization of the brain through mapping, is to figure out how regions move from one spot to the other due to the epilepsy, and where language is, and where movement is, and where taste is, and where fear is, and etc., which is very interesting. Sometimes the epileptic focus is not on the surface of the brain, is not on the cerebral cortex outside, but it's deep. It's in some inner cortical area, and instead of doing the grid of electrodes on the surface of the brain, we insert the depth electrodes within the brain. This is an X-ray of somebody who had a lot of depth electrodes implanted within the substance of the brain from both sides, again in an attempt to identify the seizure focus and to help with mapping. And these are procedures that we perform routinely, in the world of epileptology. So after we use this information to identify where speech is and movement is and other important functions, and on the other hand where the seizure focus is, then we draw a border and we tell the neurosurgeon this region has to go and this region has to stay. And often the surgical outcome is very good. Now, there is a specific type of epilepsy that I want to talk about in the remaining time, and this is its clinical manifestations. This individual is having the seizure now. He is not responsive. He is lip smacking all the time. He's fumbling with the hands. This is called purposeless or semi-purposeful behavior. And now his left hand is starting to get a little bit tense. Okay? Now, if you tell this individual during this seizure, remember the word umbrella, and you come back 5 minutes later, he will have no idea what you said. All right, and this seizure this seizure is recorded in the epilepsy monitoring unit, as you can imagine, the continuous video EEG monitoring, as part of his surgical evaluation. But if this seizure were to occur when he's in the shower, this can lead to a burn or to drowning. If this seizure occurs when he's cooking, he can hurt himself or burn the house. So these type of episodes, they can cause a huge burden on somebody's social, professional, and cognitive life because they impair the awareness completely for minutes at a time. And then all of a sudden they find themselves somewhere and they don't know how they got there, you know. So, you know, really burdensome condition. And these seizures start in the temporal lobe, which is right here behind the ear. We call it temporal lobe epilepsy. Temporal lobe epilepsy is the most common focal epilepsy and the most common reason for referral to surgical evaluation. So mesial temporal lobe epilepsy is the temporal lobe epilepsy that emerges from the deep structure that we call the hippocampus, which is also responsible for processing memories. Mesial temporal lobe epilepsy is the most common form of intractable epilepsy, as I mentioned. And what we do for these individuals is we do a lot of testing, neuropsychological testing, memory assessment, everything, in order to predict whether surgery, meaning resective surgery, just like the initial MRI that I showed you with the removed temporal lobe, is going to result in seizure freedom and is going to result in minimal or no cognitive problems. Because the biggest issue when we remove the temporal lobe in somebody who has temporal lobe epilepsy is we don't want them to sustain a significant memory decline. Remember, the structure that is the source of the seizures deep in the temporal lobe, called the hippocampus, is the one that is crucial for memory processing. So what we do, so this is, for example, somebody who has an intact MRI, and these are the hippocampi, this one and this one. This is the right hippocampus, the left hippocampus, and they both look normal. In this individual, the left hippocampus looks normal, but the right is shrunk. We call this hippocampal sclerosis. So if I want to show you the outlines of these, you can see the outline here is much smaller than this or this. When somebody has a condition like this, so that means there is a scar tissue and the hippocampus is atrophied, that means that surgical resection is indicated, and we don't expect them to sustain a significant memory deficit because they probably have already sustained that deficit due to the process itself. And if we do the surgery, there's not going to be additional trouble. However, for this individual, somebody with a normal MRI and good memory, what do we do? So this is somebody who has a normal MRI and the hippocampi look fleshy and healthy, and they have good existing memory, and they can be, you know, a highly functioning individual, a lawyer or anything. And then we tell them that if we don't operate, you're gonna continue to have seizures because you have failed a number of medications. And if we do operate, your seizures will probably stop, but you will have a significant difficulties with memory, which can be maybe as handicapping as seizures themselves. So this is the individual in whom we often do not offer epilepsy surgery as an option. So the logic we use in clinical practice is if there is a sclerotic hippocampus on the MRI and there is poor existing memory function, that means the individual is a good surgical candidate. But if the hippocampus is intact and there is good memory function, we often do not recommend surgery. So this is a very, very, very long introduction to tell you about the patient population that we are interested in our research. The first thing I'd like to say is our research started in the basic science lab with a lot of animal experiments. We figured that if we put an electrode in the brain and we stimulate a specific structure in the mouse or the rat, then the hippocampal seizures, the temporal lobe seizures that start in that structure, which is important for memory, they will be reduced by up to 90%. And then, to make a long story short, we translated these animal findings into a human trial. And the logic behind, or the hypothesis behind that trial is this is the hippocampus. So this is a dissected brain, okay? This is the front of the brain, and this is the back of the brain. And then we dissected it in this drawing in order to show you the deep structure within the temporal lobe right here behind the ear that is called the hippocampus. And this is the structure that is very important for memory, right? Now, the hippocampus is composed of cell bodies like the ones that Drs. Lamantia and Gallo showed you, but then out of these cell bodies there is a big bundle of white matter. Now, the word white matter is probably not strange by now. Myelinated processes of the neurons, myelinated means they're covered by that isolating fatty layer, which will increase the propagation speed of electrical activity. So this is the white matter here. So this white matter here is connected with almost all the cells that are within this hippocampus. What we found out in the lab, if you stimulate this, if you put an electrode in the hippocampus here or here or here or at any part of its length and you stimulate, you may actually induce a seizure. You may induce a seizure because we don't know which part we're stimulating. And when you stimulate one part, a neighboring part will have altered activity and so forth. But what we found is if you put the electrode here in the white matter, then you're activating the whole hippocampus synchronously. It's like your electrical stimulation is overriding the hippocampal activity, is telling the hippocampus you're not allowed now to fire this region or that region or that region. I'm the maestro. I'm going to tell all the cells to fire synchronously when I want. And this led to plastic changes within the hippocampus that did not affect memory at all, but actually reduced seizures significantly. So the stimulation at this stage from a single electrode placed in this white matter tract will activate the whole hippocampus synchronously. And then we did this in a number of individuals, a very short-term study, which is a proof of principle study, and we found actually that the seizure odds were reduced by 90% for up to 1 or 2 days after each 4-hour stimulation session of the white matter tract that is connected with the hippocampus. So this is a direct translation, again, from animal experiments to a human trial. And this plot shows you, so at time 0 here is when each one of these individuals, each number represents a participating individual in this study. And this line here is the time they underwent the stimulation session. And this shows you the number of seizures 2 days before and the number of seizures 2 days after. And you can tell clearly how frequent their seizures were before the stimulation and how much less frequent they became afterwards for up to 2 days, despite no changes of their medication. So, of course, when we did this study, we fixed the medication regimen because otherwise it will cause interference with the quality of the data. We fixed the medication regimen and we documented that this seizure reduction was solely due to the electrical stimulation. And now we are starting to recruit patients here at GW for a longer term trial to implant a pulse generator under the collarbone that will continuously stimulate that area and to see whether this is going to result in sustained seizure reduction without affecting memory. We got an approval from FDA to do this trial, and we will be recruiting subjects to participate in this trial very soon. The other project, which targets a similar patient population, meaning those who have temporal lobe epilepsy but who have intact memory, right? Which we will also be doing here at GW, is based on this hypothesis. Think of this as the whole hippocampus, which we saw in a previous slide. So this is the whole hippocampus. If you look at the hippocampus under the microscope, it looks like stacks of coins in a way, where each coin is a small circuit. We call it the trisynaptic pathway because it consists of 3 different fibers that communicate with one another. And we believe that these are crucial for memory processing. Now, in addition to these trisynaptic pathway or the coins, you see the hippocampus is also heavily connected by longitudinal fibers. So there are transverse fibers, okay, and there is longitudinal fibers that connect the whole thing together. We believe that due to a big number of studies, including some that we conducted, that the longitudinal fibers are extremely important for synchronization of seizure activity and propagation of seizures. So that means that if you disconnect the longitudinal fibers, there is a great chance you may abolish hippocampal seizures. But because you're preserving these if you disconnect them that way and you preserve the transverse connections, which are stacked perpendicularly to the longitudinal fibers, then you may preserve memory because these transverse connections are important for memory processing. So this is a hypothesis that is based on a lot of animal studies and some human observations. So what we did is we translated this again. So we did some experiments in the rat hippocampus that demonstrated that transection will abolish seizures, and then we published another paper to demonstrate that the disconnections will preserve memory, at least in rats. And then we took this to a clinical trial where we implanted electrodes in the hippocampus surgically, and via this knife that you see here, we started to do some disconnections. So instead of removing the whole hippocampus, we sliced it, right? Instead of, think of it as a banana. Instead of removing the whole banana, we left it in place, but we performed some disconnections that will stop the communication between the longitudinal fibers. And the outcome was also terrific. This is a busy slide, but I want you to look at these two columns here. This is the preoperative seizure frequency in 13 subjects. Look at this, 1 to 2 per month, at least 1 a day, several per day, 1 to 6 per month, etc. And most of them went to 0 seizures with follow-up periods reaching 53 months afterwards. And the ones who still had seizures, the seizures can be explained by other, like for example, this person who had several seizures per day, now she had 2 and 4 years only upon abrupt discontinuation of antiepileptic drugs. This person has a new type of epileptic seizures. That means that he probably has another focus besides the focus that we targeted in the temporal lobe and so forth. So, but okay, so here we achieved excellent seizure control outcome, but how about the memory? We found actually also that the memory was more preserved than you would expect with temporal lobectomy, with removal of the whole hippocampus. So the findings so far are extremely promising for achieving excellent seizure freedom, but with much less memory problems than the classic treatment, which is removal of the whole temporal lobe. So these are the two projects we're offering now, which are investigational but highly promising, and both aim at decreasing the seizures in temporal lobe epilepsy while preserving memory. And one is called the hippocampal transections, and the other called the deep brain stimulation. With this, I thank you very much for your attention.
Go first that time.
Okay, I think, well, thank you to all the speakers for really some wonderful presentations, and I hope we have some time for questions from the audience.
So in lieu of writing the questions down and conserving time, I also have a microphone, so if you'd like to verbally share your question, that's an option as well.
So this goes to Dr. Gallo. Has any intervention been tried in any human babies? So you mentioned some.
Can you hear me? Yeah. So, well, we have not directly tried interventions in human babies, but there are several interventions that are being tried. But one of the interesting interventions that different groups have tried is so-called enriched environment. And there is evidence in the literature that there is a significant improvement in premature babies depending on which family environment they grow. And actually there is a positive correlation between some improvement in cognitive function and the educational degree of the mother. So this has prompted some behavioral interventions and educational interventions with enriched environment. In terms of targeted interventions, it's completely, molecular targets is still completely unknown, and it's a very new field of research, and promoting growth of oligodendrocytes targeting specific molecules, so with pharmacological interventions, is still at very early stages.
And can I just follow up on
sure.
that for one thing? I mean, lest you think that the enriched environment and experience is some sort of hocus pocus, this is just basically the cell biology of the brain, because when all of us were born, our brain circuits weren't completely wired together. And the finishing of the wiring, the fine-tuning, is done through a process of actually making the cells fire electrical activity. And that influences how they finish growing their processes and actually making physical connections, so that the experience, what the baby hears, what the baby sees, how touch happens, is actually a driver of the cell biology that completes wiring the circuits. So if you enhance that experience, make it richer, make it fuller, you're actually, in a sense, giving more biological gas to putting the brain together in the best way possible.
Yeah, and in fact, I mean, if I can follow up, there is clear evidence both in humans and in animal studies that developmental myelination is a highly synchronized process that involves both electrical activity in the neurons, in the axons, as well as particular developmental processes in oligodendrocytes. And so what happens in the premature brain, and why there is this abnormalities in white matter and in cognitive functions, is that this synchronized process is disrupted at birth when these two different types of cells have to create that unique partnership that results in correct myelination. So if we disrupt that during a certain developmental, very vulnerable developmental time window, then we disrupt that particular process. And that's why our studies show that it's actually intervention has to be targeted during a specific early developmental time window in the injury. Otherwise, if we miss that developmental time window, then it's impossible to regain function.
So we've got another question here that I think really can be responded to by all our experts on the panel, but I'll start with Dr. Lamontia first. Can we expect any early impact on treating Alzheimer's disease or Parkinson's disease?
Yeah, and I actually think that actually is a great question, because probably the pathology of Alzheimer's disease and Parkinson's disease is basically the mechanisms necessary to build the brain are invoked, they're complete, you build the brain right. Then you have to maintain it, which basically is probably an adaptation of what it took to build it. It's just sort of like at home. You know, you use screwdrivers and hammers and saws to build your house, and then you have to take those tools out from time to time to keep it running. So then somehow those tools run down, they don't function properly. Now, if you wait too long, just like if you wait too long to repair the house, the damage is so great that you can't fix it. So what you want to do is you want to get there when the mechanisms have just begun to get a little bit spotty. And so these early interventions are actually likely to be much more effective. But the problem is, how do you identify who is vulnerable for the pathology? Because if you wait too long to see the actual symptoms, probably that's going to be too late to get the best therapeutic effect. And that's where genetics comes in. Because the more that we understand about the complexity, but nevertheless the predictive power of how each of our genomes, in combination with the life experiences we've had, the exposures to toxins and other drugs that we've had, how that combines to put us at risk for a particular disease. It's going to say, okay, this patient is most likely to have a really good risk of developing Parkinson's disease. And there are these therapies that can be imagined that would prevent some of the worst pathogenic processes from happening, so you never get to the point where clinically you're recognizing the extreme symptoms. Now that's in outline the process, and that's why to understand all of these disorders, not only the ones that I talked to you about that start early in life when the brain isn't built right and continue, but things like Parkinson's disease and Alzheimer's disease, where you build the brain properly and then it runs down, you still have to understand the underlying developmental process by which you put the circuit together. Identify which part of that process is maintained to keep the circuit running properly, and then figure out, well, what is it that breaks that mechanism to cause the disease? And that's a big order, but nevertheless, I think it's the outline of the approach. So the question is actually is really what I think all of us in neuroscience want to do, and it's interesting that our plan, Henry, Vittorio, and I, we've made a proposal to expand neuroscience at GW further. And what we want to do is we want to recruit investigators who are working on the pathogenesis of Parkinson's disease, Alzheimer's disease, other neurodegenerative disorders, multiple sclerosis, but actually have them focus on these developmental mechanisms that go awry, because that will also complement the group that Vittorio has put together who studies early developmental disorders, as well as the group that I've hired of investigators who study those disorders.
So actually, there's a quick follow-up to Dr. Gallo here. Is there any correlation on premature birth and adult dementias like Alzheimer's disease?
I don't think so. As far as I know, I think that there is a correlation with a lot of different neurological disorders that I mentioned earlier, but I don't think that there are studies that pinpointed directly to dementia.
Great. So to Dr. Kobayashi, so 2 questions. Does it matter where along the hippocampus one severs or slices the central cord?
What we do is, one of the pictures I showed is electrodes that are implanted within the hippocampus across its length intraoperatively. And these aim specifically at recording where the irritability is maximum. So after we record for minutes in the operating room, we figure out closest to which electrode is the abnormal electrical activity maximal, and we target that spot by the transection. So we tailor the transections according to the electrical activity that we record from the hippocampus. But in all these 13 cases, there was at least 4 cuts in the hippocampus. The number of cuts ranged between 4 and 7. So that means that probably a single cut will, it remains to be tested whether a single cut will do the same trick.
So there's a follow-up to that. So once you've severed these or sliced these areas, is there a capacity to repair or reconnect there?
This is a great question. Theoretically, when you disconnect the neural tissue, it sometimes tends to reestablish new connections, which may be aberrant connections, and theoretically may sometimes be pro-epileptic, meaning the new connections, they may form recurrent excitatory circuits that lead to seizures. This is at the theoretical level. But practically, we found that the seizures were reduced for up to 53 months, after we did that. And when we imaged the hippocampus later on in people who underwent that procedure, we found that the volume was reduced a little bit by this intervention, suggesting, if anything, atrophy rather than formation of new circuits. So we still have not examined the chronic effects of the transection under the microscope, but regardless what happens, whether it's formation of new circuits or atrophy, the clinical outcome was as desired.
So I think probably Dr. Lamantia here, with the increase in autism or its diagnosis, are you experiencing an increase in the diagnosis of DiGeorge syndrome? So
no, actually, that's a really great question, because it turns out it's the opposite. It's actually, I'm sorry, you're absolutely right, but because basically what's happening, as more kids who are picked up that have autistic spectrum diagnosis, now actually pediatricians and developmental pediatricians and pediatric psychiatrists are actually ordering genetic testing in a subset, particularly if they have craniofacial malformations. And they're actually picking up a subpopulation, about 2 or 3% actually, in some instances, in studies that have been published recently, that actually are deleted. So the answer is yes, that the increased diagnosis is actually basically bringing to attention a whole group of patients who they don't have the severe cardiovascular malformations that used to bring these kids to clinical attention and required surgical intervention to help them survive. But nevertheless, they have the deletion and they have the brain phenotype. And actually, this has been seen not only for the autistic population, but also in schizophrenia now, because if you look in the schizophrenic population, about 1% of schizophrenic patients turn out to be occult deleted. They don't have the cardiovascular phenotypes, but if you take a good look craniofacially, it turns out that they have those phenotypes.
So there's a direct follow-up here. So if schizophrenia is a manifestation of impaired cortical connectivity, why does it usually become evident in late adolescence to early adulthood? Why doesn't it show up in babies and children?
Right. And this is, again, another really great question. And there are two parts of the answer. First of all, it turns out that there have been several studies that have been done retrospectively. I think the most clever one was relied on the advent of video cam recorders, because that meant that every childhood was really, really well documented. And so what was done was they collected from a population of patients who were diagnosed with schizophrenia and age-matched controls videos of first, second, and third birthday parties, and then they had them scored independently, and they looked at the child's engagement, emotional responses, engagement with parents, engagement with other children, and they just scored them on a variety of criteria and separated them into two groups. And the concordance of the group that was not socially engaged, emotionally more labile, less likely to express extreme emotion, particularly happiness, less likely to make eye contact, had random eye movements, was the group that actually ended up being schizophrenic. So you can actually spot some behavioral predictors of schizophrenia at very, very early ages. Now, the other thing is that, well, why the really pathologic expression late in life? And this comes to things that Vittorio was telling you about that we've already discussed. And that is that in humans, this period where you can change the brain based on activity and the brain is still very malleable begins at birth, and it extends really, into late puberty, probably until about the end of teenage years, as myelination in the frontal cortex is completed, etc. And it's those processes that's why you don't want a 12-year-old to drive, but you can kind of trust a 16-year-old to drive. And so that's going on. And if you think of it this way, when that's happening, there probably are some mechanisms for the brain to adjust behavior so that it doesn't go awry, so that you don't get the symptoms of schizophrenia, the positive symptoms of hallucinations and delusions, your brain telling you, your circuits telling you there's something out there that isn't actually there. And you don't get the negative symptoms, which is the social disengagement, the paucity of language, the inability to plan cognitively a series of events. And so the idea would be that when you hit the wall of no longer having the sort of plasticity you have when your brain is developing from birth through teenage years, all of a sudden the brain can't adapt as well to alterations of function because it's not wired properly. And that lack of adaptive capacity that is inevitable as we all go from childhood to teenage to early adulthood actually brings the patient to a threshold where the brain functional abnormalities are actually noticeable and really begin to impair function in living. And so there's a two-step process. One is that there are some behavioral changes that are already present, and they reflect the disrupted construction of circuits. And then this added-on layer of plasticity helps modify that when a child is young through teenage, and then when that runs out, when the brain is really in trouble.
So, all right, great. So we're going to take one more question here and then wrap up, but if people have additional questions, they can bring it up to the panel. So let me see here. So the Alzheimer's Association care presentations indicate that diet, more vegetables, for example, and better environment can impact lesions in Alzheimer's disease. Can you comment? So I think so the role of an environment really in nervous system diseases. So I think, let's just broaden this a little bit. So Dr. Kobayashi, do you think a good environment improves epilepsy?
Eat your vegetables.
There's a huge body of literature about certain foods and herbal remedies that will reduce seizures. There is absolutely no scientific evidence that they will help or not help. In general, the role of environment in epilepsy is clear because, you see, I mentioned the admission to the epilepsy monitoring unit where patients stay for sometimes a week waiting to capture their seizures in order to figure out which part of the brain the seizures are starting from. See, some of these individuals, they can have daily seizures at baseline. When they come to the unit, they stay for 5 or 6 days with no seizures. Because the environment is less stressful. They don't have to deal with the problems at work, with the traffic, with the sleep deprivation. They sleep a lot. They're relaxed in the bed all the time, and which reflects on the seizure frequency. So we know also from students, they may have higher seizure frequency around the time of examinations when they're more sleep deprived and when they're under a lot of stress and stuff like that. So all this certainly affects the seizure frequency, but it will not cause seizures in somebody who doesn't have epilepsy. But in those who have epilepsy, the seizure frequency will be highly influenced by a number of environmental factors, specifically stress, psychological stress in general. Sleep deprivation is a known also trigger, universal trigger for seizures, and alcohol ingestion. Like these are the most well-documented, the best documented triggers: stress, sleep deprivation, and alcohol use. But other than that, there's a lot of controversies about relative contributions. Some people think caffeine helps. Some people think caffeine triggers seizures. It's controversial, and maybe it depends on specific epilepsies.
So, Dr. Gallo, can you comment on good diet and the benefits to the brain?
I think that in terms of the Alzheimer, I know that it's a very controversial field, and I don't think there is any conclusive evidence that neurodegenerative diseases such as Parkinson's, Alzheimer are particularly influenced by diet, or there is a specific link. Now, in terms of brain development, obviously there is a vast literature demonstrating that nutrition is essential for brain development, and obviously for myelin, the balance between fat intake and in the diet during development for children is very, very important. So there is obviously, it's clear there is under nourishment and effects of poor nutrition on brain development are clearly documented, and this has been true for many, many years. And actually, it is pretty well known what are the molecular and cellular mechanisms that are involved. But in terms of Alzheimer, I don't think that there is any clear. I think it would be interesting to see in the future whether when we develop maybe early markers for Alzheimer's, which is basically what Anthony was saying earlier on, can we look at these markers and follow these markers depending on a number of environmental factors, including nutrition? I mean, I think that what we need is really to have some rigorous scientific testing for this hypothesis, because otherwise it's very anecdotal saying, that person was a vegetarian all her life and had Alzheimer or did not have Alzheimer. I mean, it's really anecdotal. I think we need to have rigorous testing and markers that we can follow for these disorders throughout the lifespan.
But it's never bad to eat your vegetables. So.
And it certainly is good for cerebral vascular disease to have an appropriate, well-balanced American Heart Association or Mediterranean diet. So I think
As a
there's
non-meat eater, I can certainly go on a tangent on the benefits of not eating meat, but that's a different story.
Well, so that's fantastic. I'm just going to add a few words in summary. Thank you for coming. You know, one thing, when I hear Dr. Gallo and Dr. Lamontia talk about a molecule that they've found that correlates to either a problem in the mouse or an improvement in the mouse, that means if it's a molecule, a single one, you can find a drug to modulate that. So that's where I think the therapeutics arise one day. Maybe not tomorrow, maybe not the next decade, but at least it gives us an insight that we never had before to look at therapeutics. And then you also hear about the plasticity within the brain, that it can change, and we can impact on the pathological changes with the surgical intervention that you heard about. So, you know, I think all these outstanding neuroscientists have really come to the same kind of conclusions, that we're all looking for new and better neurotherapies into the future. So thank you very much. Appreciate it.